So in this next recording of molecular and cellular biology, we're going to address carbon. Carbon as the basis for life as we know it. So it is the element that most biomolecules are made out of. And one of the reasons carbon is a foundational element is because it's very versatile. You can build lots of different things with carbon. So it can form 4 bonds with other atoms. So go back to Honk when we think about our 4 most abundant elements in living things: hydrogen, oxygen, nitrogen, and carbon. So 1, 2, 3, 4. That 4 on the C is the 4 bonds that it can form. So it's an excellent backbone because of this tetravalence, right? So we talk about a valence shell of electrons as the outermost shell. Tetra meaning 4, so you got 4 available slots for bonding with carbons, right? So as you start to build carbon molecules, you can build different shapes very easily, bonding carbon to carbon, or carbon in this case to hydrogen in many cases, and you can create chains and shapes of various length and infinite variety, really. So these are akin to using something like a Tinker Toy, where you have a wooden hub and you can have spokes attached to that hub as a toy to build things. Here, carbon is going to take on that role. Now, all these examples that we see here are hydrocarbons. They're molecules made out of exclusively carbon and hydrogen. And this is generally where we start when we want to synthesize a biological molecule, right? So these hydrocarbons, an example of which that you may be familiar with is fat, right? And fats is another word for lipid, and a lipid is one of our classes of biomacromolecules that we'll study in just a little bit in this course. So they're very high in energy, and when we look at fats, we see these long hydrocarbon tails, just carbon and hydrogen. Now, as we study metabolism, and the TCA cycle, and how your body and most living things take food and turn it into biologically useful energy, we're going to see how we can feed fat into that system with such great outputs of energy, right? So we'll understand that later, but that's a fat molecule. Now, carbon and hydrogen are great. That's really just the foundation. Okay, so that's kind of like the steel girders in the building that sets up the frame, but then you got to build it out, right? You've got to put in the drywall, you've got to put in the floors, you've got to put in the lights, you've got to put all that stuff to make it really functional. So what we have next is a series of what we call functional groups. These are elements, small groups, small chemical groups that we add to that hydrocarbon skeleton to change its chemistry, to change its function, right? So there are 7 of them that I want you to memorize, but just to take a quick look at the impact that functional groups can have. If we look at estradiol, which is an estrogen compound, a female hormone, versus testosterone, which is a male hormone, we see that their fundamental structure is almost identical, except for what? These couple functional groups. So on the estradiol, you have the hydroxyl group hanging off the end. Right? On the testosterone, you have the carbonyl and the methyl group hanging off. And those are the only differences. But then your body will respond very differently to these 2 different molecules, only because of those 2 little functional groups. Now, I'm calling these things a hydroxyl methyl group. These are the names of the functional groups that you need to know. Right? So this is just a list. You need to memorize it. You're going to see these again and again and again, especially if you go on in biology. And so you'll become more familiar with them, but you have to start somewhere, so I want you to memorize them. Not only do you have to memorize their names, but I want you to memorize their structures as well. So these can be found in any chemistry book. You can look them up online. They're in your textbook as well. So the hydroxyl group is an OH group, very, very common. We're going to see that a lot because what it's going to add is polarity to the molecule. Okay, oxygen, remember, is still very electronegative, and it's going to draw electrons to itself. Even if it's attached to a bigger molecule, it's still going to produce that local degree of polarity, and that's going to allow molecules to interact with water well. The carbonyl group, then, is that double bond of an oxygen to a carbon. We can break these down into 2 classes based on where that carbon is, into into ketones and aldehydes. But don't worry about that. If you can recognize that carbonyl, I'm happy. Now, if you kind of combine those 2 first functional groups, you can get what's called a carboxylic acid, or a carboxyl group. This is very common. It's the end of every protein. It's acetic acid here is an example, which gives vinegar its sour taste. It's acidic. Acids are sour. But you'll also notice that that proton on the acetic acid in that carboxyl group, carboxylic acid, is acts like a conjugate acid-base pair. And in fact, proteins do act as buffers of the solution because of this and other functional groups. So our three functional groups so far then are the hydroxyl, the carbonyl, and the carboxyl group or carboxylic acid. So then we want to move on to the amino groups. So the amines, okay, involve now a new element. We're looking at nitrogen tagged on. And again, this is the other end of every protein that has an amino. And we'll talk about this when we talk about the structure of proteins. But you can see also this is going to act as a conjugate acid-base pair as well, but with a very different pKa than the carboxylic acid. But that nitrogen is going to bring different properties to the biomacromolecule. Sulfhydryl groups, now we have yet another element, a sulfur. And so this is going to be tagged on. We're going to see that this is really important because we haven't talked about it yet, but we will. Redox chemistry. So this particular functional group can be used to make bridges that are covalently bound between parts of proteins or between proteins. So we talk about cross-linking cysteines, and we'll address that later when we talk about proteins. But that's SH, that thiol, that sulfhydryl group is really important, and I want you to know it and recognize it. We have the phosphates, and the phosphates bring yet another element into play here, and that's our phosphorus. So these are going to be important because this is a group that we can actually add on to and take off of proteins to act as a trigger in many cases. We can use it to pump up the potential energy of a particular molecule, and we'll see how that works as we go through the quarter. But phosphates are our next group. That's the P4O group. And then finally, the methyl group. And the methyl group is CH3. We're going to see that we use methyl groups to control the function of DNA. And that kind of bristly carbon with the hydrogens off of it is going to have an impact on the shape of the molecule and how it, quote-unquote, feels to other molecules. So you're going to see the methyl groups and methylation, the act of adding on methyl group, or demethylation, the act of removing a methyl group from molecules, is very common in biology. Right? So I want you to commit those to memory. And again, your list is hydroxyl groups, carbonyl groups, carboxyl, amino, sulfhydryl, phosphate, and methyl groups. Make flashcards. You should be able to see them in your head when you say them. Right? That's all for now.